Okay, so in this lecture, we're going to cover chapter 22, the lymphatic system. So here are our learning objectives. So these are going to be the sections of chapter 22 that you should read and that these slides should correspond with. So focus on these objectives. I'll try to highlight which sections we are going to skip, but these sections over here should be the ones that you should read. Okay. So the first thing that we're going to talk about in this chapter, and most of this chapter that I want you to know, is the vocabulary words. You're going to learn a lot more about this in your additional courses on immunology and microbiology and all of those courses that you're going to take in your nursing classes. So focus on a lot of the definitions and the broad concepts. So when you get to those, they're going to teach you all of the details that you need to know. So the first thing we need to talk about is what is immunity. So immunity is the ability of your body to prevent any damage or disease, any kind of injury to those tissues. So how we can prevent damage and disease to our body. A pathogen is something that can cause disease. So this is a disease-causing microbe. So we'll kind of use pathogen and microbe interchangeably sometimes. And that's because it's kind of a big, broad, non-specific term for something that can cause disease or make you sick. So these can be bacteria, these can be viruses, these can be parasites. We have a lot of different things that you're susceptible to, and that's our next word. So susceptibility is the lack of resistance to a pathogen. So these disease-causing microbes are going to be able to affect you in different ways. Some bacteria and viruses you're exposed to constantly, and you've never gotten sick from them. Other ones, if you get exposed to it, it's going to make you very ill. So it depends on what your specific susceptibility to that pathogen is. How much resistance do you have or do you not have to that specific disease-causing microbe in general term? So your immunity is in two broad categories. We have two big broad categories of immunity. We have innate immunity that's present at birth. So this is your non-specific immunity that you're born with. Just things that your body naturally has developed that are going to prevent you from having any kind of infection or anything like that. Your adaptive immunity is going to be your more specific immunity. So these are the defenses that are going to be specifically targeted kind of attacks against pathogens or anything that's invaded. So if we have kind of bypassed the innate immunity and actually invaded and gotten through, we are going to be able to fight those pathogens with our adaptive immunity. So we're going to talk more about the innate and adaptive immunity in a few slides. So before we get started on talking about the actual physiology of how does all of this work, we are going to look at the anatomy of how this body system is laid out. So we are not going to have this on your lab exam. Again, there's not that much to label. We're going to start here with your bone marrow. So we have talked in detail about your bone marrow. You learned about it in the skeletal system in AMP1. We talked about it a lot in chapter 19. And we talked about how it was a location where our white blood cells, which were our kind of main actors in our lymphoid system, that's where these cells were going to develop. So bone marrow is going to be a big key player in this system, and you guys know where that is. We've talked about it, again, multiple times. We also are going to have the spleen. We've looked at the spleen a little bit when we looked at the blood chapter as well. So we're going to talk today about what the spleen does for the lymphatic system. And then we will also look at the thymus. So the thymus is a special organ that we are going to look at that's only involved in the lymphatic system, as far as we know. And it's kind of super active in kids, but as you hit puberty, really starts to stop working and do what we call atrophy or shrink. So we will see this in kids, but this will not be super present in adults. We'll have more of a remnant. So again, not much to see there. Our lymphatic system's whole purpose, right, our whole function is to drain this excess interstitial fluid. So if you remember back to our chapter on the blood and we looked at that net filtration pressure equation in our capillaries, we saw that blood was filtered out on the arterial end and it should be reabsorbed on the venous end. And so there shouldn't be much net gain of fluid in those tissues, but there's going to be a little bit. And if we have any kind of exception, like case of inflammation or anything like that, we're going to have an extra interstitial fluid to help move things along through that tissue and get rid of that debris or whatever is in there. So that is where the lymphatic vessels are going to lie. These lymphatic vessels are going to lie in between the arterial end and the venous end of your capillaries. And we're going to look at that on the next slide. So that inside of those vessels, there's going to be your lymphatic fluid, which we will look at. We will see how it transports our dietary lipids. So this is going to be kind of the buffer between the digestive tract and the bloodstream with our dietary lipids. If you remember back to chapter like 2 or 3 in your book, we said lipids don't like water. They are kind of a water repellent. So we can't just stick them right into the bloodstream. We have to have them be absorbed into our lymphatic system that has more of a lipid or fat-based plasma than the bloodstream does with its watery plasma. So those vessels are going to run in between those capillaries, picking up that extra fluid, and they're going to take it to our lymph nodes. So our lymph nodes are going to be these little sites all along different areas of your body. So you're going to see lots of them along the armpit, in the groin. You will see a lot of them going up the abdomen and the chest, and you'll see them up in the head and neck area. And if you've ever had a cold or strep throat, you've probably felt those painful little lumps behind your ear or in your neck. If you've ever got a cut on your arm, a lot of the times you'll feel the ones in your armpit. Those ones will get really inflamed if you have like a cat scratch or something on your hand. And we'll look at why that is and how they respond to these different pathogens. So all of these things together are kind of going to accomplish these goals here. Draining this excess interstitial fluid and returning it back to the body. Transporting these dietary lipids from our digestive tract to our bloodstream. And then protecting against any pathogens that have gotten through the rest of the immune kind of organs. Any of those other defenses, those innate responses that we're going to talk about. So here is a picture of those lymphatic capillaries. So again, they're just going to be these vessels. They're indicated here in green that are going to lie between the arterial end and the venous end of our capillary beds in our tissues. These are going to be large vessels. They're going to be very permeable, but they're only going to allow things to kind of enter them. Nothing's really going to be able to leave. So once things enter these lymphatic vessels, they're kind of trapped in here. They get stuck. Again, we're going to have these special capillaries that are in the intestinal tract that carry lipids. Ooh. So we will talk about these when we get to the digestive tract. But they are called lacteals. So lacteals are special lymphatic capillaries that carry these lipids in our lymphatic vessels. So this is a special kind of fluid or vessel. So the chyle is the fluid that's in there. So this chyle is this white lymphatic plasma. It's inside of our lacteals, and that's what's going to physically allow those dietary lipids to get carried away and along the lymphatic vessels and then eventually return to circulation. So these lymphatic vessels, again, they're all going to run just like our veins do. So these are going to run just like our veins do, forming trunks. So all the vessels that are going to exit the lymph nodes are going to meet and form lymphatic trunks. These lymphatic trunks are then going to dump into what we call the thoracic duct and the right lymphatic duct. So the thoracic duct is going to collect all of the lymphatic fluid from the entire left side of the body and then the lower right side. So the thoracic duct is really going to receive all of that fluid from the whole left side of the body and the lower right portion. So the majority of our lymphatic fluid is going to flow through the thoracic duct. The first portion of the thoracic duct that's going to receive lymphatic fluid is going to be called the cisterna chyli. So the cisterna chyli is the first portion of that vessel that is going to get the lymphatic fluid. The upper right part of the body is going to be drained by our right lymphatic duct. So the upper right part of the body is going to be drained by our right lymphatic duct, and that's going to return again to circulation. So these are going to dump into the venous system and it's going to return blood to the heart. Again, right near the junction of the internal jugular and subclavian veins. Okay, so that was the actual structure of our vessels. Again, they're going to run just like your capillaries do. They're going to collect into bigger vessels called lymphatic ducts. And so that lymphatic fluid, again, is going to come from that excess interstitial fluid. So here you see our heart, you see our arteries coming out to the arterial end of the capillary bed. You see our venous end of the capillary bed and veins returning that blood to the right side of the heart. So those lymphatic capillaries are going to kind of fall in the middle of those like we saw, and they're going to reabsorb all of that interstitial fluid that's there. They're going to send that fluid to lymph nodes that are going to filter that fluid and allow that whatever is inside of those lymphatic fluid to come in contact with our immune system cells. So we'll look at that here in a few slides. So as that fluid is then sent out of the lymph node after it's come in contact with those immune cells, it's going to form those lymphatic trunks or lymphatic ducts, and those are going to have valves in them. So those lymphatic vessels have valves just like veins. They're going to help prevent that backflow of fluid because, again, lymphatic fluid is going to be just like the venous system. It is going to move via those same mechanisms, that respiratory pump that we looked at and the skeletal muscle pump that we looked at when we looked at how blood returned to the heart and what was responsible for venous return. Those two factors are also going to be responsible for the lymphatic fluid return. So again, these are just going to work the same way. The skeletal muscle pump and the respiratory pump are going to help push lymphatic fluid upwards against gravity, and these valves are going to help prevent our backflow. So here you can kind of see our whole schematic of how these are going to work. So when we look at all of our fluid, it's going to flow into specific organs. So we're going to have primary lymphoid organs, and we're going to have secondary lymphoid organs. Primary lymphoid organs are where our immune cells are going to develop. So that's going to be our bone marrow and our thymus. So we'll see our red bone marrow and our thymus will be responsible for various cell development, and we'll look at those in general. And then we're going to have our secondary lymphoid organs, and this is actually where the immune response occurs. So this is where our immune system cell, is actually going to meet up with that pathogen. So this is where the germs are going to meet our cells that are going to hopefully fight them off. So this is going to occur, again, in those lymph nodes. So we're going to talk about those in a little bit more detail. It's also going to occur in the spleen. And then our lymphoid nodules. These guys aren't actually considered organs because they lack a capsule. So tissues that lack a capsule are just kind of considered tissues rather than organs. They kind of don't meet the criteria. So we'll look at those lymphoid nodules in this chapter and a little bit in the digestive tract as well, because they're very important there. Whoop. There we go. So our first lymphoid organ that we're going to talk about is one of our primary lymphoid organs, and this is our thymus. So our thymus is going to be the small kind of organ that sits right here in the anterior chest. It's going to sit right on top of the heart. So here's our heart covered in that pericardium that we talked about in chapter 20. So here it is sitting all nice and protected, and the thymus is going to sit right on top of it between the right and the left lung. You can see it's going to run from the sternum to about the fourth costal cartilage. So you can kind of feel that on yourself from that sternal notch. Count down 4 of those spaces between your ribs, and that's about where the thymus would lie. Again, this has to be encapsulated because we consider it an organ. So it's going to have a capsule, and we are going to separate this into different lobes by what we call trabeculae. So trabeculae are just like partitions that are going to separate this organ into different lobules. The whole function of the thymus is to help T cells develop. So the T cell in thymus, or the T in T cell stands for thymus. So this is where our T cells come from the thymus. So T cells are going to develop here a lot. Again, all the way up until puberty. Once puberty starts, this organ is going to start to atrophy. T cells are still going to be here, but there's going to be a lot less of them. And you can see as you get older, you're going to form more of these thymic corpuscles. So you can see one here. It's this lighter area out here, and you can see one here. So these are biopsies of the thymus. And these thymic corpuscles are going to be areas where T cells can no longer develop and proliferate. These are just areas filled with keratin, which is like a protein that you have in your skin. We don't know if these have any other function other than they're just non-active parts of the thymus. So maybe you will see this in upcoming research during your lifetime, but for right now, we just think that these are just kind of inactive areas of the thymus. And as you get older, more and more of these thymic corpuscles form and less. Okay, sorry about that. I don't know if you could have seen that or not, but no worries. It's all good. So again, we don't know what these thymic corpuscles do other than these just indicate areas of atrophy of the T cell's ability to proliferate. And this is going to cause the whole thymus to shrink up eventually, and we'll just have what we call a remnant left over. All right. So the next structure of our lymphatic system that we are going to look at is our lymph nodes. So we have about 600 to 1,000 lymph nodes in the body, depending on the person, depending on just the anatomical differences. Lymph nodes are all, again, going to have a capsule. So they're going to be wrapped and kind of isolated and just as their own structure. They're going to have these trabeculae that form these little lobules and kind of section them into little partitions. And then they're going to have an outside cortex and an inner medulla. And I like to think about this like a pie. So a pie has a crust all the way around it, right? With a pie with a crust on the bottom and the top. And then the inside where all the filling is, that's like the medulla. So the outside of the pie, that's the crust, is like the cortex. And the inside, that's the middle, is like the medulla. So that's what's happening all around this structure, because this would be a 3D structure. We're just looking at it flat right now. So it would have an outer cortex all the way around it, and then the inner part would be the medulla. So the cortex is going to be on the outside, medulla is on the middle, is in the middle. So the afferent lymphatic vessels are going to flow into this lymph node. So these are bringing fluid from the tissues into this lymph node. So again, they're going to be located all over the body. So they're just going to be kind of local area. So again, why if you have a cut on your arm, sometimes the ones in your armpit are large, because those are the lymph nodes that are draining that area. So all of these different afferent, we call them afferent, lymphatic vessels are bringing fluid into the lymph node. They're going to filter that fluid through the cortex and the medulla. And you can see these little boxes are showing you this part of the cortex is going to have these specific immune system cells. The inner cortex will have these guys. Medulla will have these. And again, going to be important depending on what infection you're fighting, or if you have a part of your lymph node that's not working, you have a cell that's inactive. Again, lots of different disease states and possibilities that you guys will learn. But for now, all we really need to know is that we're going to bring in fluid from all of the local tissues via afferent vessels. It's going to get filtered through kind of layers of lymph node and get past different immune system cells. So on level 1, basically in our outer cortex, right, if we have B cells and macrophages, so we're going to filter out some of our bacteria and things here. Then we're going to filter some other ones in the inner cortex. And we're going to filter other pathogens or microbes in the medulla. And again, the whole point of this is just to have lots of different cells for these pathogens to go past, right? We want to have every opportunity to be able to filter them out and identify them. I like the analogy that the lymph node is kind of like when you go through customs. When you come back into the United States from another country, they make sure that you don't have anything that you're not supposed to. So no like illegal fruits or vegetables or animals or drugs or anything like that. They have multiple different ways of searching you, right? They have those little sniffer dogs. Maybe they're going to x-ray your bag. Maybe somebody's going to open it up and start opening up your things and going through them. They have multiple security checkpoints to check for different things that would be inside of someone's luggage or something coming back into the United States. And the same going into other countries, right? You go through customs that way too. But that's what this lymph node is like. It's taking in all of these different afferent vessels, bringing fluid in, filtering it past various stages of cells that are going to detect different things, and then sending that fluid that's now been kind of deactivated or kind of neutralized out via our afferent lymphatic vessels. So this fluid has all been checked. It's all been dealt with by these immune cells. If we saw a pathogen, we hit the alarm button, said, hey, we should look for this guy, right? And so we'll talk about that in a little bit more detail. But that's how these lymph nodes work. They're going to function, filter any of our lymphatic fluid and remove anything that's not supposed to be there. So again, you see lots of macrophages on different levels, and they're going to filter out anything that's not supposed to be there. So again, you don't need to know what's in what area. You just need to understand in general, how does a lymph node work? So here we can see kind of the macroscopic view of a lymph node. So if we went in and we did surgery to remove these, this is what it would look like for the surgeon. This isn't a cadaver, so it would look slightly different, but this is in general how it would look. And then if we biopsied this lymph node and we looked at the histology, this is what we would see. Here we can see that capsule. Here we can see our outer cortex. Here we can see some medulla. So again, we would be able to biopsy this lymph node and see, is there anything abnormal? It doesn't look like it could be cancerous. Lymph nodes are often the times the way that cancer spreads or metastasizes. So we need to evaluate lymph nodes to evaluate the spread of cancer as well as disease state. So the rule of thumb is usually if it's a lymph node that's enlarged and it hurts, it's usually active. That means that it's doing its immune system function. If it's enlarged and it doesn't hurt and it's kind of really rock hard, those are the ones we recommend people get biopsied, because those ones are oftentimes, not often, but they can be cancerous. And a lot of times we can detect cancer this way before we would see other symptoms. Some people get a weird lump and they're like, hey, I went to the doctor and had this lump. A lot of times that lump is a lymph node that the cancer has spread to rather than the original tumor. So again, then we'll be able to biopsy that cancerous lymph node if we find one and figure out what the cancer is if we can't figure it out initially. Or in the case of breast cancer and several other cancers like skin cancer, we can take and inject radioactivity around the actual tumor that we know where it is. To say we know where the surgeon's going to cut, we would inject our radioactivity around that right underneath the skin, so it was taken up by the lymphatic system. And then we would image that area and we would watch that radioactivity be taken up from underneath the skin into the local lymph nodes. And then we would send that patient into surgery, and the surgeon would take those lymph nodes out, the ones that were most likely to spread cancer or become metastasize the cancer, because they were draining that local area. So lymph nodes, use this little guy a lot for a lot of different things. Again, it can be really good at detecting viruses and bacteria and pathogens, but it can also spread cancer. So again, we will see this if you do an oncology unit in nursing. You will talk about lymph nodes a lot. All right, so here's our kind of last lymphatic organ that we need to talk about. I said we weren't going to talk about bone marrow. We've handled it enough. We talked about the myeloid and the lymphoid stem cells. You guys know bone marrow is where the B cells come from. And when we get to the B cells in a few slides, we'll talk about that. But that's pretty much all we need to know today. So our last anatomical organ here of our lymphatic system is going to be our spleen. So our spleen, again, in a capsule, all organs are, almost all, and it is in our upper left quadrant. So again, you're not going to really feel this or see this organ. He's kind of small, he's kind of tucked in. So when you're kind of palpating or feeling the abdomen on your patients, unless this is enlarged, you probably won't feel it. But it is going to have two different areas. So on the inside, we're going to have white pulp and we're going to have red pulp. When blood flow comes into this splenic artery, so we looked at this artery, it's going to be on your lab exam, or I think it is on your list at least. So your splenic artery is going to bring this blood into your spleen. The blood is then going to flow first through your white pulp. Inside our white pulp area, so that's going to be this area of here on the histology, this white pulp area is going to have all of those immune cells in it, B cells, T cells, macrophages, and they are going to, again, perform the kind of those same functions they did in the lymph node in the spleen, but now with blood. We already said the spleen was a great filter. It filtered out all those old, damaged, not usable red blood cells. So we are also just going to stick some immune system cells there and have, while it's filtering those things out, we're going to have it filter for pathogens as well. So that's what the white pulp's going to do. We're going to filter for those pathogens first. Then the blood's going to, after it's passed that test, it's going to go flow through that red pulp. And that's where the filtering of those red blood cells is going to happen. So you guys know all those steps of what's happening in the red pulp. We just are adding the white pulp to the story now. So blood flows in through the splenic artery, breaks up into all these little, you see these small arterioles, and it's going to send blood through the white pulp to be filtered by the white blood cells, and then to the red pulp where we're going to filter out any old or damaged red blood cells. And then all of that blood that's been filtered is going to be able to leave via our splenic vein. So that's what our spleen does. Again, just a big filter. A lot of filtering going on in the lymphatic system. Oh, we didn't talk about lymphoid nodules. So our non-organ lymphoid tissue. These lymphoid nodules are just masses of lymphoid tissue. A lot of them you're going to see what we call mucosal associated lymphoid tissue, or MALT. This is going to be in a lot of your mucous membranes, so digestive tract, urinary tract, genital tract, and respiratory tract. You're going to see lots of MALT. So we will talk about MALT again. We are going to specifically talk and touch on the Peyer's patches in the ileum, and so we'll look at those in detail when we get to the digestive chapter. We are also going to talk about the tonsils when we get to the respiratory system later this week. We are going to look at the 5 different types of tonsils that we have. So we are going to have the pharyngeal tonsil, which we call the adenoid, which again is going to be in the nasopharynx. We're going to have those 2 tonsils that when you open your mouth and you say ah, you have, those are called the palatine tonsils if you haven't had them removed. And then we will have 2 lingual tonsils, and these will be at the base of your tongue. And again, we will look at these in the respiratory system lab and lecture. So those are all of our structures for the immune system. Now we are going to look at how does the immune system actually work? What is our function, and how does this work to kind of prevent any kind of infection, and what do we do if a pathogen gets into the body? So innate immunity, or again, is an immunity that we are born with. So this is present at birth. We are going to have 2 lines of defense, a first line of defense and a second line of defense that are part of our innate immunity. The first line of defense is going to be your skin and your mucous membranes. Again, your skin is going to have that thick epidermis that you guys looked at in A&P1. And as long as that skin stays intact, meaning not cut open or anything like that, it's going to be really good at keeping things out of your body. So this is why we make sure that we keep our hands nice and clean when we work in healthcare. Try to make sure you don't have any cuts on them, things like that, because this is going to be a really good barrier for you to help protect yourself. Another first line of defense is our mucous membranes. Again, our mucous membranes are going to have multiple components, mainly one being mucus, right? Mucus is like that thick stuff when you think of snot or anything like that, that is going to kind of collect anything that comes into those openings to your body. So your eyes, your nose, your mouth, they're all going to be able to make mucus that's going to be able to get any kind of dirt or debris out of them. You can see hair in mucous membranes, like in your nose. So you know that when you have snot in your nose, right? If you go to like a campfire or anything, you can see that you get smoke up your nose. It's going to get trapped in that mucus and on those hairs. And when you blow your nose, you're going to be able to see that kind of stuff, those particulate matter like stuck in there. You're also going to see cilia, which are small microscopic hairs at mucous membranes. These are going to actually be able to beat, like move, and they're going to be able to move things forward and out. So this is why you cough things up and out. The cilia are going to help move all of your respiratory mucus and things like that up and out. Mucous membranes are also going to have various enzymes. So depending on if it is your eyes or your nose or your mouth, you're going to have different enzymes in there that are going to prevent different bacteria from being able to enter. So if they come in, that enzyme is going to go and just degrade them immediately. You're also going to have a lot of fluid in those areas. These different fluids we'll talk about when we get to these areas. Saliva in the digestive tract, gastric juice in the digestive tract, vaginal secretions when we get to the reproductive system. But how all of these fluids have different components and different properties that make them really good at expelling whatever would be in that area. So saliva has lots of things that prevent from bacteria that gets on food from coming into your body. Again, gastric juice, really good at breaking down things that would come in with food. So we will look at these when we get to those various chapters as well. For now, just understand that mucous membranes are one of our first lines, is our first line of defense, and these are the components that make it our first line of defense. Our second line of defense is going to be our internal defenses. So these internal defenses include things like antimicrobial substances, natural killer cells and phagocytes, and the inflammatory pathway causing inflammation, and then the formation of fever. So that's what we're going to look at in detail now. We're going to look at each of these second line of defense kind of properties in detail. So the first one is going to be our antimicrobial substances. These antimicrobial substances are just located in different parts of the body, all over the body, a lot of the times in the bloodstream, that are going to be able to prevent different things from infecting you. So these are just natural chemicals that your body makes that help defend against certain pathogens. A large group of these kind of antimicrobial substances or chemicals are going to be called interferons or INFs. These are going to be what any kind of infected cell, like a lymphocyte, a macrophage, or a fibroblast. So if these cells get infected, they're going to release these interferons, and interferons are going to signal the neighboring cells, hey, there's a virus on the loose. It's going to come into your house. Don't let it replicate. And so that's what's going to happen. So these uninfected cells are now going to be able to prevent this viral replication because of a signal from their neighbor to signal, hey, get ready. There's going to be a virus that's going to try to infect you. So these interferons are kind of antiviral chemicals. They're going to help prevent viral replication. Another kind of pathway that leads to antimicrobial substances, is going to be complement. So complement is a big, long cascade of proteins. We're not going to learn it in detail, but we'll look at a picture. So complement is going to be this big cascade of proteins that eventually will end with cytolysis or cell rupture of any of our microbes. It's going to increase phagocytosis, and it's going to help inflammation. So complement, again, is just a kind of a sequence of events that's going to help promote all of those things in the case of pathogens entering the body. So we'll look at complement in a little bit more, in a few more slides. So iron binding proteins. Iron binding proteins, again, are going to be just innate proteins that are made in your body. Their goal is to bind iron and prevent bacteria from being able to use it. So bacteria need iron to grow. If we decrease the amount of iron that's available for them inside your body, you're going to be able to decrease their rate of growth. So that's good. We want to be able to prevent this bacterial infection from getting worse. So these iron binding proteins are going to help limit how much bacteria can grow by limiting how much iron is available to them. We've already looked at all of these in chapter 19. We talked about hemoglobin and how it had iron at the center of that big ring structure to hold oxygen. And then when we broke it down, we said if we release that iron from the hemoglobin molecule, we had to couple it with transferrin in the bloodstream because we could not have iron be alone, right? It had to have a chaperone. And this is why, because bacteria will come along and steal it. If iron's just loose in the blood, the bacteria is going to come and steal it and go crazy growing. If it's bound to one of these larger molecules, the bacteria is not going to be able to take that iron, and it's not going to be able to grow. So those are our iron binding proteins. Our antimicrobial proteins are going to be our these are kind of short proteins, and they have this really kind of broad spectrum of activity. These activate dendritic cells and mast cells, which are going to help present antigen. They're going to help us alert the body that we have a problem. Some examples of these are dermacidin and defensins. Again, I would not ask you those specifically. It's just this is a broad group of categories, and they're going to, again, have a broad spectrum of activity depending on where they're located and what cells are releasing them. All right. So we are looking at our list here of our innate immunity, our second line of defense. So we just finished looking at our antimicrobial substances. Now we're going to look at our natural killer cells and phagocytes. So the natural killer cells and the phagocytes are going to work together kind of like a 1-2 hit of our pathogen that's invading. So pathogens, again, are going to be two places when they get in your body. They're going to be free floating around, or they're going to get inside of a cell and try to hijack it and take it over. They're going to infect it and try to grow more bacteria, or if it's a virus, viruses, right, they can't replicate on their own. So they're going to steal all your cells' ability to replicate, and they're going to start using it for themselves. So we don't want that to happen. So we are going to have to be able to take out pathogens or microbes, as they use in this example. Again, same, just broad category, something that can infect you. We have to be able to take them out in two places. We have to be able to take them out inside the cell, and we have to be able to take them out when they're loose and floating around. So we have two ways to do this just innately. We're going to talk about ways that we do this adaptively as well. So innately, the first thing that's going to happen is that you're going to have these cells floating around in your bloodstream. They're going to be in all your lymphatic organs. Again, these are natural killer cells are going to come in first, and then the phagocytes are going to come in second. The natural killer cells are going to be able to identify cells that have an abnormal plasma membrane. So they are going to have some protein on their plasma membrane that says, hey, I'm infected. This isn't right. There's some way that the natural killer cell is going to be just floating around looking at them and say, that's not right. That cell is infected. When that natural killer cell finds that infected cell, it binds to it, and it does two things. It releases one chemical called perforin. Perforin causes perforations or holes. It pokes little holes in all of the cell membrane. After it pokes those holes, it releases a second chemical called granzymes. This is kind of a group of enzymes. These granzymes are going to enter through those holes, and they're going to digest everything inside that cell, leading to apoptosis or cell death. So, these perforin and granzymes are going to kill that cell. As that cell dies, now that infected cell is dead, but some microbes might be able to get away. Some of the microbes can be released and would be able to infect other cells in that area or travel around the body and infect other cells. So again, we have to be able to take out these microbes in two ways. Once when they're inside the cell, and the ones that escape that are floating around. So we are going to kill these ones that escaped this cell and escaped death by natural killer cells and kill them with our phagocytes. So again, these phagocytes are just these cells that are be able to do this job called phagocytosis. We have a lot of different names for these cells depending on where they're located. Our phagocytes are gonna perform phagocytosis in 5 steps. The first one is called chemotaxis. Chemotaxis is the phagocyte releasing chemicals that will attract the microbe towards it. So this is like if you walk past a bakery or a restaurant or something that's making really good food, or like cookies or something, and you can smell them and you're like, oh, I have to go in there and I have to have that cookie, or I want to go in and get that pie, right? That's exactly what this chemotaxis, that's what these chemicals are doing for this microbe. They are drawing it in and attracting it to the phagocyte. Once this microbe's close enough to this phagocyte, it's going to stick to it. So step 2 is adherence. The microbe and the phagocyte are going to stick together. Once they're stuck together, the phagocyte's going to be able to ingest it, so bring it in. So just like you ingest food, this phagocyte ingests a microbe. Once ingested, that phagosome, which is a little pod that is holding this microbe or pathogen, is going to bind with the organelle called a lysosome. So you guys looked at lysosomes when you talked about the cell in A&P1. Lysosomes contain lots of digestive enzymes that break things down. So step 4 is digestion. Those enzymes from the lysosome are going to digest that pathogen. They're eventually going to break the whole thing down, killing that entire pathogen and rendering it harmless, leaving everything that's left over in what we call a residual body. This residual body just kind of chills in these phagocytes until they are digested and expelled through the body, right? All cells have a lifetime, so this phagocyte's not going to live forever. So these residual bodies are just going to hang out in there until we get rid of that whole cell. So again, natural killer cells are going to come in first, do their job. Then the phagocytes are going to come in and eat up anything that was left over or anything floating around. Another one of our second lines of defense is inflammation. So this is when there's any kind of tissue damage. When there's any kind of tissue damage or injury, we need to get cells out to that area to help repair that tissue. So inflammation is the way we're going to do that. The first step is going to be vasodilation, which again is going to be increasing the size of the vessel and the permeability of the vessel walls. So we can have step 2, which we talked about in your blood chapter, I believe, where you had immigration of those white blood cells out into our tissues and help go fight any of these pathogens. So here you see the phagocytes have immigrated out of this vasodilated blood vessel and are performing chemotaxis, our first step of phagocytosis, to try to prevent these microbes from entering the bloodstream at all. And then we'll be able to go ahead and repair this tissue, which you guys talked about when you did the skin chapter and you did deep versus superficial wound repair. So this is kind of the steps right before that. Symptoms of inflammation. So I'm sure you will chart these. I'm sure you have felt these if you've ever gotten a cut or hurt yourself at all. It's going to hurt, right? You're going to actually feel pain. It's going to be red because we're increasing the permeability of this blood vessel. And it's so when we do that, fluid is going to float out and accumulate in that tissue. It's going to cause that tissue to swell, and that swelling is going to cause immobility or loss of function of that area. As we have all that fluid and swelling there, it's going to put off heat. So it's going to feel warm to the touch. So it will be warm, red, it's going to be swollen, it's not going to be able to move well, and it's going to hurt. Those are our symptoms of inflammation. Inflammation can be acute, meaning it's mild, it's local, and it's short-lived. This inflammation is going to kind of be handled by our neutrophils. They are going to be the phagocytes involved in this step. If inflammation is slow to develop and chronic and long-lasting, that's chronic inflammation. And we're going to see monocytes and macrophages involved with this. And you will see lots of different acute and chronic inflammatory conditions in your future courses for sure. All right, so here's table 22.1 summarizing all of those innate defenses for you. So you can come and check this table and look at all of those different innate defenses, our first line and our second line of defense. All right, so now we're going to look at our adaptive immunity. This is the immunity that develops over time after exposure to a specific antigen. What is an antigen? An antigen is anything that provokes an immune response. So anything that causes an immune response. This can be foreign or yourself. This is where autoimmune diseases come in. So it's really important to keep the definition of antigen general. It's not always foreign. It's not always just a bacteria or a virus. It can actually be your own body proteins. So anything that causes an immune response is an antigen. So when we look at our adaptive immunity, we're going to have two main principles: specificity and memory. Adaptive immune responses are more specific. These are targeted attacks. We are only attacking the strep throat bacteria, or we are only attacking the Staph aureus bacteria. We are looking at a specific molecule. We are targeting a specific thing on that pathogen. And adaptive immune responses are also faster. The adaptive immune system is what lets our immune system make memory cells that remember specific antigens. So this may be why you had strep throat a lot as a kiddo, and maybe you haven't had it in 10, 15, 20 years, because your body has developed a lot of these memory cells, and if you're exposed to it again, you don't get sick. Maybe you don't even feel any symptoms. So our adaptive immune response involves our B cells and our T cells. Our B cells, again, develop in our bone marrow, and our T cells develop in our thymus. So we are going to kind of have two different upgrades of B cells, and we're going to have two types of T cells as well. So we're going to have these general B cells, right? They're mature, they're ready to go, they're active. They have these antigen receptors on them, so they're going to be able to float around the bloodstream and detect antigens. When they detect an antigen that binds to them and provokes that immune response, they are going to get activated and form two special types of upgraded B cells. They're going to form a plasma cell, which is a little antibody factory. This B cell now knows which antigen has bound to it and says make antibodies that attack just this. So again, if it was the strep throat bacteria that was present in your body, it bound to this B cell. This B cell would make a bunch of plasma cells that made lots of antibodies that were specific to that strep throat bacteria. We would also make memory B cells so we remembered for next time and we can mount a faster, stronger response. So B cells are responsible for what we call antibody-mediated immunity. They make antibodies. These, again, are those little seek and destroy missiles that are very targeted and go and destroy whatever antigen they are told to. Whatever antigen they are made to take out, that's what they take out. So we looked at these guys when we did the ABO blood grouping, or ABO blood typing. So what about these T cells? T cells, again, develop in the thymus. There are two types. We have what we call helper T cells, and we have what we call cytotoxic or killer T cells. Helper T cells are going to have a protein on them that is called CD4. So this is just like a little marker that is coming out of the cell membrane. Your cells actually, if you try to think of a golf ball, so our cells are 3D. They look more like a golf ball than these pictures. And if you imagine each one of those little divots that's in a golf ball had a name. It was a special protein and we knew what its name was and what it did. That's more what your cells look like, realistically. Tons and tons of proteins. Now, instead of those being divots, they would be proteins sticking outwards. So it's tons and tons of proteins sticking out all over this 3D ball, basically. And so one of those proteins on this cell, on a helper T cell, is going to be the CD4 protein. Cell differentiation marker 4 or something like that is what it's called. Not super important. But again, when you see this CD4 cell, you want to think of helper T, because you're going to learn a lot of diseases like HIV and AIDS that affect the CD4 T cells, and a lot of treatments, especially for AIDS, that is going to help these CD4 T cells and work on this cell. So more on that later when you get to those courses. For right now, I just want you to understand that our helper T cell is going to also be called a CD4 cell because it has a CD4 protein on its cell, on the cell surface. Okay. So the CD8 cells then, on the other hand, are going to have the CD8 marker. So they're going to have a protein that is a clustered or cell differentiation marker 8. So these are going to just have the CD8 protein. CD4 helper T cells are helpers. They help activate the killer T cells or cytotoxic T cells, and they help activate B cells. So helper Ts help activate killer T cells and B cells. Killer T cells then do exactly what they say. They are cytotoxic. They are killer T cells. They're going to go and they are going to take out any intracellular pathogens. So I said we were going to talk about ways to kill pathogens specifically and non-specifically. So we looked at those natural killer cells that are kind of the innate counterpart of this. These cytotoxic T cells are going to be much more targeted and more of a specific killing a specific intracellular pathogen. And again, you will also see these in cancers and transplant rejection and things like that. So again, important to understand your CD8 cells and your CD4 cells because you will learn more about these later. Our CD8 or our cytotoxic T cells, we say, are responsible for our cell-mediated immunity. So B cells did antibody-mediated immunity. T cells are responsible for cell-mediated immunity. Okay, so how do we know how to make more B and T cells? We do this via a process called clonal selection. So when we see one of those bad guys, a pathogen, on our B cells or T cells that's floating around in our plasma, they are going to get exposed to that antigen, that strep throat antigen. Well, we only need cells that are going to fight off that strep throat antigen. We don't need to make cells that are going to fight any other bacteria or virus or parasite right at that time. We have a targeted opponent. We are going to fight that strep throat bacteria, and we are going to form a clone army. So again, we know what the antibody is that we need. We know what the cytotoxic T cell is that we need, and we're going to select for that one, and we are going to clone a bunch of it. This is going to occur again in these secondary lymphoid organs, and this is why you feel those swollen, painful lymph nodes. This clonal selection process, this making of these B and T cells that are targeted to your specific pathogen or antigen at that time, these are going to be activated, and this is going to occur in these lymph nodes, and so these lymph nodes are going to swell because they're active, and it's going to hurt a little bit. So you're going to feel those. Again, we're going to have two types of cells be formed during our clonal selection process. effector cells, so cells who do action. So these are those helper T cells, cytotoxic T cells, and the plasma cells that make the antibodies. And then we're going to have memory cells that aren't going to do anything right now, but they are going to remember this pathogen for later. And these cells, again, are going to have a long lifespan, some of them up to decades. So you can have a lot of good immunity or prevention against infection from a lot of different pathogens for a long amount of time based on the amount of memory cells you have. Okay, so let's look a little bit at these antigens and receptors. So again, antigen is anything that provokes an immune response. This can be foreign, like a bacteria or virus, or a self-protein. Immunogenicity is the ability to cause that immune response by stimulating production of those antibodies or B and T cells. So immunogenicity is the ability of that pathogen to be able to stimulate that B or T cell response. Now, how does this antigen and antibody bind? So an antigen, again, is just this protein or molecule that you see here, and it's going to have a lot of these markers on its surface. The specific area that the immune system is going to recognize is called the epitope. So just these three little codes right here, this is going to be our epitope. This is the part of the antigen that is going to cause the immune response. So the rest of this molecule the body's fine with. It just doesn't like this part. It's like, hey, that epitope, that means that we need to attack it. So most antigens, most pathogens are going to have epitopes directly on their surface. Your body's going to recognize it as foreign and go, we're going to attack that. Now, some things might be too small to actually cause an immune response alone. We call these things haptens. So haptens are molecules that are too small to spark their own immune response. But when combined with another molecule, they form a large complex that the immune system can react to. And a good example of this is poison ivy. So poison ivy is a plant native to PA, right? Very common to get poison ivy. It's got those 3 little shiny leaves on it. Those leaves are covered in this urushiol oil, not important. The oil, when that gets on your skin, is a hapten. This molecule is too small itself to cause that itchy, kind of bubbly poison ivy rash by itself. But when combined with some of the proteins that are in your skin, can cause that rash because your immune system can now see it and react to it. And that's what's causing that effect. So this is why we say if you've been exposed to poison ivy or when you come in from outside walking through grass, you should wash off with it, just rinse with cold water for like 5 minutes. Flush the area, try to get all that oil off your skin. Because once you start washing with warm water or soap, you're going to help push that oil into your skin to combine with that protein and make this response. Again, some people are going to be more susceptible than others, but haptens are just molecules that are too small to provoke an immune response alone, but when combined with something else, the immune system can now see them and react to them. Another big vocabulary word that we talk about when we look at antigen receptors on our B and T cells. So how are these B and T cells able to bind these zillion different antigens and know which one is which one's which pathogen? And this is done by a process called genetic recombination. So if you take immunology or micro or a genetics class, you'll learn a lot more about this. But basically, it's just the actual genes, so the DNA of the B and T cell receptors are going to be able to recombine, kind of switcheroo pieces and parts in that DNA molecule to make new combinations and make new receptors. And that's what's going to allow for that receptor diversity and the ability for these B and T cells to be able to bind a lot of different types of antigen. Okay, last little kind of cell surface marker that we are going to talk about on our cells. So again, on a human cell, we said we were going to have all of these proteins sticking out of it. It's going to look like a golf ball with flags coming out of every one of those divots that we're naming. We're identifying all of those little spots. One of these proteins is called the major histocompatibility complex, the MHC molecule. MHC molecules are unique to each individual person except identical twins. These MHC molecules can come in 2 classes, we call them, basically 2 types. Class 1, on all human cells except red blood cells. These say this is my cell, this is going to identify all of your cells. You are going to have all of your own MHC class 1 on all of your cells, except for red blood cells. And remember, that's just, we said red blood cells are kind of odd. They're not really even cells. And then our MHC class 2 are going to be on the receptor, or are going to be on the cell membranes of all of what we call our antigen presenting cells. So our antigen presenting cells are going to present antigen. So they are going to float around the body and present this antigen and expose any of the other cells in that area and saying, hey, this is the invader. This is who we are looking for. This is who you should be fighting. So how do we deal with these antigens? So we're going to briefly look at this process so we can briefly discuss MHC class 1 and 2, and that's it. Again, I'm not going to ask you all the steps of this picture. But again, we have 2 types of antigens. Exogenous antigens, the antigens that are in the plasma, and then endogenous antigens, antigens that are inside of our cells. So when this antigen or pathogen, whoever's out here hanging out outside the cell, again, it's going to be taken up by a phagocyte and it's going to undergo phagocytosis. We're going to break down that molecule. When we do that, we are going to take a piece of that antigen and we are going to present it on our antigen presenting cell. So our antigen presenting cells are our phagocytes. As they are floating around, cleaning up all of those debris, germs, pathogens, they're going to be able to take a piece of that pathogen and present it on their cell surface. So they can identify, hey, everybody look out for this guy. This is the antigen that we are trying to fight. And same thing's going to happen with the endogenous antigen. This antigen, again, is going to be digested at some point within our side of our cell, and it's going to be presented on an MHC class 1 molecule. Again, letting all the immune system cells know that this cell is now infected. So this body cell is going to let the immune cells know, hey, I'm infected. Please come take care of that. And so that's what your MHC1 and MHC class 2 molecules are. They are just the molecules that are going to present that piece of antigen on the outside of the cell. Whether it's saying, hey, the phagocyte that's saying, hey, I found this floating in the plasma, everybody be on alert. Or it's a cell that's actually gotten infected and it's saying, hey, I'm infected. Please come take care of this antigen. Okay, and again, not a lot in detail about those. I'm not going to go in detail. I just want you to understand what MHC1 and MHC2 are so that when you hear more about them in microbiology and immunology and so on, you are aware of that they are talking about cell surface receptors that present antigens in various conditions. Okay. So cytokines. Cytokines, again, in general, we are not going to really talk too much about cytokines. These are just small kind of hormones that are going to be able to stimulate and inhibit cell functions. So these are going to be able to turn on and off immune system cells. So sometimes we're going to say, hey, we need more immune cells. We have an infection. And sometimes we're going to say, okay, we got that pathogen handled. Can you control that? Can you get those? We don't need any more immune system cells right now. When we're going to inhibit their function. Here's an example of some of them. Again, I'm not going to ask you anything about this. This is just so if you ever hear interleukin or tumor necrosis factor or something like that you understand that it's a cytokine and that is a chemical in the body that is going to signal cell function. It's either going to turn that cell on or turn something off inside of that cell. Again, no specifics about these except this interferon, which we already talked about on the other slide. All right. So this is a slide that you definitely need to know. Your antibodies or immunoglobulins. So you're going to talk a lot about these in your other classes as well. So we are going to kind of understand what they are and what they do. Antibodies, again, are those structures that are released from our plasma cells that target specific antigens. These are those seek and destroy missiles. We looked at these when we looked at the ABO blood grouping. So if you were type A blood, you had anti-B antibody in your bloodstream. So if you ever got type B blood in your bloodstream, it would go and attack it. So what do antibodies do? Well, they're going to neutralize other antibodies. Not that big of a deal. They're going to immobilize bacteria. They're going to agglutinate antigens. They're going to activate that complement pathway. And they're going to enhance phagocytosis. So really helpful and important. Here are the 5 types. They spell out the word gamed if you like acronyms. The E and the D would be switched on most to least. Your book is listing these, and you have the most IgG antibodies at any time, just like at a normal body state, and you would have the least IgE. So we're going to look at what conditions would cause those to be elevated. So here's your IgG antibody. Immunoglobulin, that's where the little I and G come from, and then capital G. So immunoglobulin G, IgG, most abundant. This antibody is floating around in the blood, in the lymphatic fluid, all over. It is going to be targeted to specific bacteria or viruses. Again, it can enhance phagocytosis, can activate lots of different things. Kind of a non-specific response, just alerting the immune system. This is the only type of antibodies that babies are born with. This crosses the placenta from mom to baby. This is the only type of antibody that can cross the placenta. IgG. All right, IgA antibodies. These are found at mucous membranes and in secretions. These are going to be what are in your tears and your saliva to help prevent any kind of infection. So we can see that this is a little bit of a bigger shape. And again, this is just going to be in your secretions, sweat, tears, saliva, and breast milk. So this is going to be the second type of antibody that babies usually get. So they get IgG from the placenta, that passes through the placenta, and they'll get IgA from breast milk until they can form the rest of these. IgM is going to be a big antibody. You see it looks like a snowflake. It's huge. This is going to be in your bloodstream. This is going to help with this immobilization of bacteria and agglutination of antigens. This is going to wall things off. This is what those anti-A and anti-B antibodies of your ABO blood group are. And you actually saw these guys in action when you watched them agglutinate. So you watched that watched this, and we saw pictures of it, I guess. We didn't really watch it, but we saw pictures of this in our blood lab. So this is the type of antibody that is responsible for agglutination and immobilization of our bacteria. IgD are on B cells. We don't really know what this does. Honestly, I don't really have a good answer for you, and I don't know if anyone does. Hopefully in your career, maybe you'll see some more research on IgD. Right now, I can't tell you too much about them. IgE, however, is going to be a very specific antibody, and we are going to see this in allergic and hypersensitivity reactions. So asthma, allergies, allergy testing, parasitic worm infection. So again, if you live in areas of the world where parasitic worm infections are common, or you go to the beach in the summertime, right? Hookworm right through your foot, very common. So IgE is going to be the elevated, we would see elevated IgEs. If we looked at your patient's blood sample, we'd also see those eosinophils be elevated, right? So we are going to start to put together the pieces of this puzzle. So that is our IgE antibody. Definitely want to know these guys for my class and for your future immuno and micro courses. Okay, as promised, a small picture of complement that is really not that big of a deal. I don't need you to know complement. Complement is a very detailed cascade of proteins, which is these things that look like, I think, Tylenol that they're breaking in half. These are different proteins that when cleaved into various parts, so you see we have C3 gets cut into C3b and C3a. And those pieces do different things. It's a big old cascade of proteins that ends with 3 effects. 1, we're going to enhance phagocytosis. Again, this complement pathway is going to make these microbes want to get eaten up by this cell more. 2, we're going to increase inflammation. So this complement pathway is going to activate histamine, which is one of those chemicals that it causes. So this complement pathway is going to cause histamine release. Histamine is going to cause vasodilation, which is going to help with inflammation. Complement is also going to help form cytolysis because these last couple little activated proteins are going to be like little needles almost, and they're going to be injected into the plasma membrane of an infected cell, and you can see they put a big hole in it. It's just it's like popping a balloon. We are going to stick this needle, which is all these proteins that compound together, forming a membrane attack complex. So this complex of proteins is going to attack the cell membrane. It's like popping a balloon. We are going to lyse that cell, and everything that was in that cell is going to come out, and that cell is going to die. So again, complement is this complicated cascade of proteins that ends with increasing immune system responses. All right, here's a small graph from your book on memory and the immune response. So you can see here that we have our primary response in this darker colored area and the secondary response in the lighter colored area. You can see after the first exposure, we'll have our primary response be primarily IgM antibodies, and there's not going to be very many of them. And it's going to take a little bit for those IgGs to really kind of catch up. You see, we're really a good couple days before we can form those IgGs after our first exposure. So that was our primary response. After that, during our primary response, we make memory cells. We said we made memory B cells and T cells. So those memory B cells, if you get exposed to that pathogen again, are going to be able to make those antibodies a lot faster and a lot easier now. And so now you're going to see this huge up spike of these IgGs in response to this pathogen now. And it's going to be able to be almost right after that initial or that second exposure. So you are going to be able to form this great secondary response. It's going to be faster and stronger, and hopefully you will get sick for a less amount of time and with less severe symptoms, or maybe not at all. Okay, 2 more topics, and then we are done with this lecture. So, and another important topic that we need to talk about with these cells, our immune system cells, our B and our T cells, is something called self-recognition. So, we need your cells of your immune system to recognize your cells as your own cells, but not bind and destroy them. So, in a 2-step process, we are going to approve your B and T cell for release. We are going to say, okay, you have to pass 2 tests before you leave this area. Before you get out of the bone marrow or the thymus, you have to pass these 2 tests. One is the test of positive selection. Can they recognize self proteins? So you see here we are in the thymus. Does this immature T cell recognize the self MHC protein? If yes, it's passed the positive selection test. It knows that that is your cell. If no, it can't tell that that's your cell, and it might attack it. That's bad. Nope, can't. That one's got to die. We cannot allow that one to be released. So positive selection allows for us to be able to say that your cells can successfully recognize your own MHC molecules. I know that this is my cell, and I'm not going to attack it. Negative selection ensures that you don't respond to those self proteins. So, is this T cell going to recognize any proteins that are your own protein? If it can, and if that T cell receptor is going to be able to bind that protein, we got to get rid of it. If no, it passes the test. We want it to not be able to bind your own proteins. So again, positive selection allows that we make sure that we recognize our own cells. Negative selection makes sure that we don't bind to and kill our own proteins. And we're going to do this with B cell or T cells up here, and then our B cells here. Again, thymus and bone marrow. All right, and our last topic is going to be, how do we get this immunity? These are really highly tested. I will definitely test you on these. These will probably, I've seen people have test questions about these. I've heard of nursing questions about these. I've actually heard of NCLEX questions about these randomly. So these 4 methods of acquiring immunity randomly pop up again. So definitely note card these, or however you like to study, but get these 4 things down. So we can acquire immunity either naturally or artificially. And that process can be active, meaning your cells make the immune response, or passive, where you don't actually have to do any work. So we'll look at each one of these individually. Naturally acquired active immunity is when you're exposed to that pathogen, it stimulates that response, and you make your own antibodies and do your own work. So, example here, the one we've been talking about this whole PowerPoint, strep throat infection. You get exposed to the strep throat pathogen, it stimulates those B and T cells, and you make that immune response, that cell-mediated immunity and that antibody-mediated immunity yourself. Naturally acquired active immunity. You naturally acquired that infection, you actively did something about it. Naturally acquired passive immunity. So, naturally acquired, again, we're going to send you naturally made antibodies, but you're not going to have to do anything. So we're going to send preformed antibodies one person to another. And we looked at this when we saw the antibodies pass across the placenta or via breast milk. These antibodies are already preformed. Mom did all the work. The fetus, the baby doesn't have to do anything. We are just giving these right from the mom to the baby via the placenta or breast milk. So that is our naturally acquired passive immunity. Artificially acquired active immunity. So artificially acquired, meaning we gave you an antigen to induce immunity. So you still had to do the work. Your body still had to do the work, but you didn't get just exposed to this antigen naturally. So an example is going to be a vaccine. So like your tetanus vaccine. So if you've gotten a tetanus vaccine, we gave you some non-pathogenic portion of that tetanus, and we injected it to you, and it produced an immune response. You formed memory cells. So next time your body sees that tetanus, if you step on something when you're outside or cut yourself, whatever, next time your body sees tetanus, it is not going to cause an immune response. Or hopefully your body's going to be able to handle it. So artificially acquired active immunity, our example are vaccines. And then we are going to have artificially acquired passive immunity. Again, we are going to inject someone with preformed antibodies this time. So not the pathogen where they have to do the work themselves. We are going to give them antibodies that are already made to a pathogen. So this is an example of rabies. So if you get bit by an animal that we suspect to have rabies or we don't know had rabies, or we don't know if it had a rabies shot, we can give preformed antibodies to rabies and prevent you from hopefully having any kind of infection from that bite. So that is our artificially acquired passive immunity. All right. So here's a summary again on table 22.5 of all those cells and how they work together to perform your adaptive immune system responses. So again, stick to your learning objectives for studying this one. Remember to take your end of chapter assessment. And that was everything for chapter 22.